still very relevant. And that's why I'm just gonna leave it there for those of you that want to rewatch on this boring topic, or if you missed it, that's available there. There is a drug table here, Word document. It's nothing for you to memorize or anything, but sort of put things into a table form. Feel free to take a look, but if you don't like things like grouping this way, then do not worry about it. It gets busy, or it gets busy. And and lastly, I've already posted my review questions. Now remember, the exam for this course is Tuesday after we come back from Thanksgiving break. So here I have the review questions broken down by topics in terms of antifungals, influenza, and also HIV. Now we don't have a whole lot of drug for influenza, right? And just basically the two mechanisms, and for antifungal it's also not too bad. The HIV, yes, we're gonna look at a lot of different individual drugs, but in terms of mechanisms here, I'm more focusing on drug classes, right? Every single drug that is in that drug class, I'm gonna just lump it all together for one mechanisms, like how we talk about. There are some notable facts of particular drugs that I'm going to point out in the lectures and also reiterated on the review study guide. Um, so, so that's something you want to focus on, right? So this study guide is very, very precise of what I'm going to ask on exam 4. So please pay attention. The minimum that you need to prepare for my exam questions on exam 4 is complete this guide. Now, fortunately, we still have Monday after we come back and do a quick review. So, and if there's anything you need to catch me, definitely that Monday before exam, catch me and take out the opportunity to ask everything that you need to ask. All right. So that's the housekeeping thing. And I think by this time, most that are coming in is coming in again. Not recording this lecture because the material is the same from the past 2 years, and the old recordings are already posted in case you need to review that. Let me do the share screen again. All right. So today we are looking at the HIV drug pharmacology and some relevant medicinal chemistry. Now, there's a lot of info that is being presented on the slide, and some of the infos I'm gonna just fly a little bit quickly. Not that they are not important, but I think it's just there are a lot of subtle things that uh, I don't want you to I don't want to make you to memorize for exams. And I'm sure I make you to memorize and you're gonna forget it, and then it's a waste of time. So I want to focus on more of the big picture. Like I said, the review questions really, is gonna be helpful. So we're going to look at a brief overview of the HIV viral cycle and look at the mechanisms of these drugs and pointing out some of the notable features of particular drugs. In terms of the HIV viral cycle, now we have a lecture in immunology, right? Looking at some of the deeper science in HIV. So I'm not gonna reiterate that again in this lecture. So here it's just a very quick review of the HIV virus life cycle. Remember, HIV is the virus, AIDS is the disease. And I think recently I've heard that people are talking about renaming AIDS for whatever reasons. They keep renaming things these days. But it's not that official yet. But just so you know, maybe one day we don't call AIDS anymore. But HIV, the virus, is not gonna change the name. Human immunodeficiency uh, virus. All right, so this virus, and in terms of its structure, it is an enveloped virus. So it is covered with a specific glycoproteins on its surface, namely the GP120 and GP41. Now, these two viral proteins both play a role in attaching to the CD4 receptors that is on the CD4 T cells. And along with the attachment, it also requires co-receptor, which are the chemokine receptors, CCL5 and CCL4, for internalization, meaning for entry, right? So remember, there's that two part. There's the attachment and there's that entry. So we always think of it almost like a continuous step, but there's this small one and two. Okay, one and two. One is attached, second is entry, internalization. So those co-receptors are important, right? Are important and needed, right? Are required for internalization. So now, once it goes in there, it goes through very similar viral cycle steps that we talked about yesterday. The membrane is gonna fuse the viral envelope. It's gonna fuse with the host cell membrane and releasing the nucleocapsid, and then going through this uncoating process. All the content that is contained inside the nucleocapsid is going to be released. What are the contents? RNA, because HIV is an RNA virus. And also what's packaged together is like enzyme called reverse transcriptase. And HIV must be reverse transcribed. Its RNA must be reverse transcribed into DNA, right? To carry on, the further down steps of the viral cycle. And the reverse transcriptase is very unique to the virus, and we don't have that reverse transcriptase in our cells, so that's why it's packaged along in the HIV viral particle. Now, once the RNA is reverse transcribed, it will form the double-stranded DNA. Now, this double-stranded DNA is going to further down integrate or fuse into our chromosome or into our DNA. So this is a very unique features of these retrovirus, and this integration is done by an enzyme called integrase. Now, once it's inside the host nucleus, the host then would transcribe. Okay, once it read that DNA, it's going to transcribe that DNA into messenger RNA. And this viral-related messenger RNA is going to be translated into a protoprotein or pre-protein that is related to all these viral proteins. And this premature viral protein further down is going to be processed by specific HIV protease into mature proteins that will be finally everything will be come to the pictures and be assembled, and lastly the virus will bud off again and then move on to infect another cell. But now I want to reiterate or point out this viral cycle to you guys is that, well, hey, this is the picture. This is actually the golden picture, and we're gonna look at snapshot of that when we talk about the drug classes. Many steps in this viral cycle can be interfered or inhibited by specific drug classes. And in here, we see that we have something called the CCL5 can be antagonized, CD4 can be antagonized. So these drug classes are going to affect the entry, right, or binding, or entry, or fusions, the steps of the viral cycle. And we have a big class of drugs, two classes in precise, something called NRTI or NNRTI that block reverse transcriptions. And we also have drugs that are called integrase inhibitor that blocked integrations. And lastly, but not least, we have drugs called protease inhibitors that prevent the processing of the premature viral proteins. What it means is that that protein is going to be just a big chunk and not getting chopped into the small pieces. So we have multiple steps. So apparently it's pretty good. But unfortunately, like all drugs, the virus can all develop resistance to one class. And you will learn about that in the clinical lecture tomorrow from Dr. Bailey, is that none of these drugs are used alone. It's always HIV is always a cocktail treatment, okay, meaning that there's multiple HIV drugs put together in a combinations and given in a specific regimens for patients and for maintenance therapy, right, to keep the viral low. So by utilizing different mechanisms together, you decrease the chance of resistance and maximize therapeutic effect, right? So there's a lot of intros staying on this page, but hopefully that set the grounds for everyone to look at things more specifically. Now we're moving on to different drug classes, and I'll point out some specific note on certain drugs. Now, the first drug class, I kind of put it together, is we call it fusion and entry inhibitors. Now, fusion and entries, when I put it together, but there's some subtle difference, okay? Like I mentioned earlier on, fusion inhibitors actually prevent the binding between the virus and the CD4 T cells. And they do it by either binding to the GP41 or binding to CD4. So it's that initial binding. And entry inhibitors, it's similar, but they bind to their targets after the virus is already attached. So that's why I try to emphasize that subtle differences between fusion or binding and then entry. So that's that attachment and then go in, right? So remember that's that two difference. And entry inhibitors bind to that after the attach. Now they can either bind to the CD4 to prevent it from interacting with the CCL5, which is required for entry. They can also bind directly to CCL5, right? So that's also an option there. So what are the drugs? Right, here are the few drugs that are in this fusion entry. So for fusion inhibitors, we have enfuratide and CCL5 antagonists. We have maraviroc and CD4 post-attachment HIV inhibitors. We have a monoclonal antibodies. So let's look at it with a little bit more detail, just slightly more. Now, a lot of the things here, I put it just for completeness. Especially some of these pharmacokinetics things. I'm not going to ask those on exam, but but like I said, it's more for your FYI information there. The enfuratide, yes, that's a peptide. So the name, and with the tide usually is a peptide. So here's the drug molecule just for your reference. What it does, it can block HIV entry to the host cell by binding to the GP41 glycoprotein that is on the virus itself, right, and the subunit. So by binding to it, it blocked the attachment to the CD4. All right, so that is that fusion inhibitor. It is an IV drug. You can probably guess that it's a peptide. There's no way that you can just eat it, right? It's just by oral. Now, mutation can happen. You can change that GP41 a little bit, and then that will prevent this drug from binding. Now, once you go to the entry inhibitors, all right, so here we have a small molecule drug, maraviroc. Now, this one is a CCL5 antagonist. So it binds to CCL5. Now, unfortunately, not all HIV use CCL5. Some would use CXCL4. Some use both. So Maraviroc has a huge limitation that it only work against HIV strains that use CCL5 exclusively, and those that use other co-receptors, it will not work. It will not work. So it's very specific, and that's why the use is quite limited, and you have to do all of these testings. It's again, it's a good thing is that it is oral drug, so administration is a little bit more easier. Again, mutations can occur, and usually due to driving the proliferation, because if you are blocking those that the viruses that use CCL5, then the virus that is like kind of like a selection pressure, those that use CXCL4 will proliferate more, and that becomes an issue. So that so alone is not useful. Another entry inhibitors we have here, I mentioned early on, is this monoclonal antibody, ipilimumab. And now you don't need to worry about these very specific name too much. Like I said, but if I would put those on, I will put both name on it. So this one is a little bit different. A monoclonal antibody that it's binding to CD4. Like CD4. Remember that co-receptor that is required to interact with CD4. Now, if this binds to CD4, if theoretically it blocks universally, doesn't matter if it uses CCL5 or CX